Welcome to the Help Me video for the reproduction and inheritance test. Quite a tricky test in places this. So question 2 starts off with plant sexual reproduction. Most flowering plants are adapted for pollination by wind or insects. So it's a general term. Define the term pollination. And so what we're really interested in is the transfer of something, and where does it go from, the male part of the plant, and where does it go to in the female flower. So what's transferred, where's it going from in the male to where's it going to in the female. Name the structure X in the diagram. So X is actually the middle, but it's all of this oval shape in there, so the kind of inside bag bit of that in the female part of the flower. Draw a line on the diagram to indicate the route of growth of a pollen tube through the carpel to structure X. So this is all the carpel, and there's the pollen grain, and so you just need to work out now where the female gamete is located in here and where this tube is going to grow. So it grows down, but then it's actually going to grow through a certain area of the female part, and you need to name that as well, or know where it is and be able to draw it. So just one mark for that. And then, oh, hang on. We said this was about plant reproduction, and it suddenly starts talking about an animal sperm entering the cytoplasm of the egg. So this is an egg, and these are the various different stages of a sperm entering the egg, or at least part of it. Name the part of the reproductive tract in which these events take place. So where is this egg in the female reproductive system when fertilization takes place? Use the information in the diagram to explain the role of Y. So Y is this area here on the front of the sperm, and it says use the information in the diagram. So we need to say, there's no mark for actually saying what it is, so it's the acrosome. And you need to say what's inside that acrosome, and then it's trying to give you a clue here as to what's happening. The acrosome is causing this process here to go on. So tell us what's being produced there, the job that it's doing, and where this is, and it actually gives you the name there. And now quite a difficult thing to do. State two similarities between this process here and the process of, it's got you to draw this tube down here. So similarities between those two processes, and you might notice that this process here that we've just asked you about, what's being produced, well that happens in both. That could be a similarity. The fact that we've got this structure here forming, and that's similar to the structure that you drew down here. What travels through this tube will be the same thing that travels through the tube there. The way, the kind of direction, there's a kind of long word that describes how the growth of this tube is, the direction of growth is stimulated by chemicals. There's a word that describes that, and the fact that this is a cell with a cell membrane. However, then something happens to that membrane in both of these situations. So it might seem quite difficult at first, but there's quite a few similarities, and you just need two of them. Then we move on to question 7 in this test about the ABO blood grouping system. In the spec you don't need to know a lot about multiple alleles, and that's okay, because it's telling you here everything that you need to know. And it's saying that there are 3 different alleles that produce something or don't produce anything at all. And here are some genotypes. You'll notice here with the genotype there's 2 copies of the alleles. There's always 2 alleles present in an adult. And then that's our phenotype there, our blood group. So explain why people with the genotype IA, IB have the blood group AB. And this is a question of, here we are, so what we're trying to get you to is look at this information here. That's what it's telling you in the question. The AB there gives AB there. But in the normal kind of Mendelian genetics, we might expect one of them to be dominant over the other and just a single phenotype being expressed. But here we've got a kind of a mixture, as it were, of the phenotypes, and there's a word that describes where both of these phenotypes are being expressed. So both of these alleles are being expressed, and the clues up here telling you what's being produced. Explain how meiosis results in people with the blood group AB producing 2 different types of gamete with respect to the gene for the ABO blood groups. This might seem quite complicated, but really it means you're going to tell us a little bit about meiosis, and you know all about meiosis. And here this information is telling you that our genotype is heterozygous. So we've got the IA and the IB. So this is heterozygous. And we're producing different types of gamete. So gametes that are not identical. So you know that if you are heterozygous for something, the possible gametes that you can produce might be either of those alleles, because only one of the alleles is going into a gamete. So which one is it? Well, that would be completely random. So use your meiosis information to talk about how many of the alleles are going into the gamete and why it could be either of those. Part B. In one family, 4 children each have a different blood group. And anyone who's actually done lots of kind of blood group questions will sort of recognise this question. It's a common question. Their mother is group A and their father is group B. Yes, yeah, this sounds exactly like 100 questions before. Complete the genetic diagram to show how this is possible. So if we've got 4 different blood groups, then you need to work out what parents might give those. And a clue here is, if the parents are blood group A, they could be capital A, but would that be able to give all 4 different blood groups? And hopefully you realise that one of these children is going to be blood group O. So one of these children is going to be blood group O, and what's the only way you can get blood group O is that if these alleles, so if it's homozygous for O, and so that will then, you think, well, you get an O from the mother, an O from the father, and then that really completes what these blood groups are going to be. So there's your clue there. And we've got some data. So 120 in total for the number of children, and we've got the different blood groups there. Our system would suggest that we're going to get a 1-to-1 to 1-to-1 ratio. But those don't look all the same to me. And so we're thinking, well, are they near enough 1-to-1 to 1-to-1 to 1 or not? What would we expect in that normal ratio? Well, we'd expect 120 divided by 4 is 30. So we would actually expect 30 of each of those in a perfect 1-to-1 to 1 ratio. So is that near enough? Well, who knows? The chi-squared will tell us. And so these columns here are observed, have gone in there, are expected, are our exact 1-to-1 ratios, and then we are just taking one away from the other, squaring it, and then dividing it by our expected again. And then this just means add them all up. So you'll get a number here for chi-squared. This will be a number, and it asks you, saying the critical value for chi-squared, the degrees of freedom at 0.05 probability is 7.82. So our critical value is 7.82. So you've got your chi-squared number and you're comparing it to the critical value. And there's different ways to remember it. The way that I remember it is that the null hypothesis is that there's no difference or no significant difference between the observed and the expected. And if our chi-squared is bigger than our critical value, we can reject that null hypothesis. So there is something else going on. If chi-squared is under our critical value, then we have to accept the null hypothesis and accept it's just down to chance. There's nothing significant deviation from our 1-to-1 to 1 ratio. So that's just a bit of maths there. And then we've got question 3, a squirting cucumber. Google it. And 3 types of plants. This is all very strange. Plants with male flowers, plants with female flowers, and hermaphrodite plants with both. And whether it's male, female or hermaphrodite is just determined by a single gene with 3 alleles. So again, you need to remember how many alleles will be present. And there'll be 2 copies of an allele. So it's going to be AA, so it's definitely going to be AA, but then what have we got up there? And the starting question is okay, the genotype of a female squirting cucumber. So this is just testing whether you understand about the dominant and recessive. And if you've got a recessive trait, then in order for that to be expressed in the phenotype, there's certain conditions that you need for that. So they need to be both recessive alleles be present in the genotype. It would have to be homozygous for the female allele. And so you just need to pick out the female allele and make it homozygous, and that'll be the answer for that. Part 2 is an absolute stinker. It's a bit strange to begin with because you explain why a male squirting cucumber cannot have the genotype D. And you think, well, okay, well, hang on. D is dominant, so if it was D, then it would be male. Yeah, okay, well, what's wrong with that? I can't see any combination of these if we added anything else to it it's definitely dominant. And the problem with it is, well, where did it get those Ds from? And the clue is this must have had a male parent, or a parent with a male flower, and a parent with a female flower. And as it stands at the moment, this would have been produced by two males, which is impossible. Two males cannot reproduce. So it must have got one from another phenotype. So then you can now work out the combinations of what might give a male phenotype, but not with that genotype. It'll definitely have one of those present, but it's dominant. The other one might be different. Complete the genetic diagram to show how it's possible for a cross between two hermaphrodite plants to produce female offspring. The clue here is that the hermaphrodite allele, so the plus, is dominant over the female D. So the hermaphrodite is dominant over the female. And if we are producing a female offspring, the female, well, that's recessive, and the female phenotype is only expressed if the genotype is homozygous for that recessive. And so that female offspring must be two of the recessives. And so you need to think what hermaphrodite parents will be able to give that. So it must have got a little d from one of them and a little d from the other, which hopefully will give you enough clue there. And then we're into female hormone cycles. Starts okay, and it's talking about FSH and LH with 5 marks. You should hoover up these 5 marks. Okay, and just start talking about FSH. Say what it does, what it acts on, what it causes, that sort of information. And again, with LH, tell us, when it's produced, what happens, and does it stimulate something to do anything else? So just dump your notes about FSH and LH there. The estrous cycle of female sheep can be synchronized by giving them low doses of progesterone. Then when treatment stops, the sheep come into estrus a short time later. Explain why low doses of progesterone prevent estrus in sheep. Well, you need to think about the effects of progesterone on another hormone, the method by which it works. So if you give the sheep progesterone, it stops another hormone from being released. It limits another hormone, and then that will have, so something else won't happen. The key point in the menstrual cycle won't happen. So you need to tell us what that is. And then as soon as you stop this progesterone treatment, the sheep suddenly come into estrus there. So if you stop the progesterone, then that stops inhibiting another hormone, and so that other hormone will then be released and will have its normal effect. So identify the hormone that is not inhibited any longer, and so when you get a surge in that other hormone, what's the key event in the estrus? So what will happen? So hopefully that first page isn't too bad. Then we've got some data. This is the number of sheep giving birth. Synchronized sheep. Let's paint a mental picture of that. Number of sheep giving birth, and that's the days after the first lamb. So just as soon as one's born, then we just record how many how many sheep give birth. And we've got a control group here. What they did was they didn't give these the progesterone. So the synchronized sheep had the sponge inserted with the progesterone soaked in, and the control sheep presumably just had a sponge with liquid, but not actually the progesterone. Using information from the graph. So we're gonna have to read these numbers in the graph. Suggest the advantage to a farmer of synchronizing estrus cycles in sheep. The farmer doesn't just do it for fun. There must be some benefits to having all of these cycles exactly in time with each other. And firstly, you possibly need to think about how these sheep become pregnant in the first place. Well, you need a male sheep, the ram, and quite often there's only a few rams that need to inseminate a lot of ewes, which is the female sheep, and they can do this in quite an efficient way if all of the ewes, the females, come into estrus in time with each other. So this could then happen over a few days rather than a few weeks. So it's keeping the ram in the field just for a shorter time period rather than a longer time period. So you could try and summarize that sort of answer. And also the logistics of keeping these lambs at the same stage is more efficient. You can prepare for this or try and come up with an answer how you might be better prepared or organised, or an efficient use of time. It's quite a woolly answer, really, if you forgive my pun. Sorry. Estimate the length of the estrus cycling sheep. This is better. Explain how you arrived at your answer. So don't just give a number, tell us how you got it. So the length of the cycle. So presumably the cycle, the peaks in these births would correspond with the ovulation peaks in the cycle. That's the presumption that we're making there. So just read off the graph where the first peak is, and then read off the graph where you think the second peak is, and that's your number, and tell us how you've arrived at that. So give us your reasoning. Then just a little bit on the end, describe how the controlled group of sheep should have been treated. I think I gave you a little bit of this answer. So tell us what they got and what they didn't get, and tell us why you need a control experiment, really. And you've done that in your plan, in your planning, in your practicals when you tested that skill. Okay, good luck in your corrections.